welcome to this online talk tonight. Thank you very much for joining us. We're here with Professor Lynne Boddy, who's going to tell us all about the mysterious hidden kingdom. So I'm very excited to pass over to Lynne.
Well, thank you very much, and thank you very much for inviting me. Let's see if I can share screen okay. This could be the first tricky bit, couldn't it? The next tricky bit will probably be when we find out that I can't progress the slides, which occasionally happens, it seems, nowadays for no known reason. Okay. Right. So, the mysterious hidden kingdom. I'm imagining that this is probably the sort of thing that you're expecting to see. It's the fruit body of a fungus. It's the fly agaric, Amanita muscaria. But actually, it is just the fruit body. And of course, it's equivalent to the flowers and fruits of flowering plants. And as with plants, you know that plants, there's much more to them just than the flowers or fruits, the leaves and the roots and the shoots, etc. And there's much more to the fungus than just the fruit body. These clouds of dust-like particles that you can see floating around beneath, those are the spores produced by the fungus. They're equivalent to the seeds of flowering plants. And usually they drop within a few meters of the fruit body, but of course, occasionally they can spread very, very much further than that. Now, the spores are made on the gills, which you can see here, and I'm sure that you're used to seeing these types of fungus fruit body, and indeed I expect you've seen lots of brackets like this. In this case, the spores are made in these tubes, which you find on the lower side of the bracket. But there are other sorts of fruit bodies too. This is the fruit body of a fungus, and you could be forgiven for thinking that it's the bark just peeled back, but it isn't. That is the fruit body of a fungus. These too are fruit bodies. In this case, the spores are made on the tips of these antler-like projections. And here we can see similar things in a flower-rich hay meadow. These are our wax caps fungi here, and these are other fungi such as earth tongues, which you only find in these pristine habitats. We don't see them in much of our grassland, which is so-called improved, in inverted commas, grassland, the improved bit meaning that loads of fertilizers have been added and just perhaps a couple of species of quick-growing grasses have been planted. In Britain, we're pretty well off for flower-rich hay meadows, in other words, unimproved pasture, depending on your definition, or unimproved grassland. So we get these wax caps, earth tongues, and other threatened species there. And talking of threatened species, and also to illustrate a different sort of fruit body, these are tooth fungi. This is Hericium coralloides and Hericium erinaceus, and you can see these pendulous projections. It's on these that the spores of these fungi are produced, and these again are threatened species. Here's another threatened species, the rare oak polypore. Well, actually, it's gone back. Oh, how did that do that? It's gone back to its original name, Bugglossoporus quercinus. I must change that. I guess you wouldn't describe this as being a charismatic fungus, and perhaps that's one of the problems which fungi have from a conservation perspective. Many of them are not particularly charismatic, although I think you'll probably agree that these are. So Bugglossoporus quercinus, Britain is probably the world's stronghold for this fungus, and indeed its epicenter really is Windsor Great Park, where we have these wonderful ancient trees which are host to this fungus. Here are some other peculiarities which you may have seen or perhaps may not. In this case, we've got one fungus fruiting on top of another one. There's one here fruiting on this. And these are sometimes commonly called piggyback fungi. They are fungi which are parasitic on other fungi. Another example here with a different type, a different phylum of fungi there fruiting on what is called a basidiomycete. In fact, everything that I've shown you so far, they belong to the phylum Basidiomycota or basidiomycete which is less formal. Here are some more examples, different things again. These are jelly fungi. And you can see where they get their name, although this one looks really rather crispy. This is witch's butter, Exidia glandulosa. But if this gets dampened up, again, it will become very gelatinous and very rapidly start to produce its spores. This is the ear fungus, Honiola auricula judei. And another one, which sometimes given the same name as this. I said this is witch's butter. This one is sometimes called witch's butter. More commonly, the common name is the yellow brain fungus. Then there are the puffballs or stomach fungi, sac fungi, they're sometimes called. Well, they shouldn't be called sac fungi because that refers to a different type of fungus too. But these stomach fungi are, as you guess, sort of stomach shaped, but the spores this time are produced inside of this container, and they then puff out. They puff out when there's a drop of rain or a twig or an animal's foot presses on them. These also fit in the group which is sometimes called stomach fungi. These are the stink horns, and they develop from what are called eggs, in inverted commas, underground and rapidly erupt. And a gelatinous mass is formed on the heads of these stink horns, and the spores are produced here. This gelatinous mass smells like rotten flesh, and you would have smelt it in the woodlands. People sometimes think that it's the smell of foxes or something like that, but it's a smell of rotting flesh, and it attracts flies to it. They wander around and feed on it, and the sticky spores will stick to their bodies and be spread elsewhere. So this is the stinkhorn fungus, Phallus impudicus. This is a relative, the dog stinkhorn, Mutinus caninus. And so spores often spread by wind, which we saw earlier, and in this case they spread with insect vectors. Now, as I said, those are the basidiomycete fungi, one phylum. There are several more phyla, varies depending on which taxonomist you speak to. But overall, this is a huge kingdom. Kingdom fungi perhaps is a kingdom of 3, 4, 5 million species. I showed you all those photographs to start with to show you some of the variety just within one phylum. And I'll show you just a few other examples from another phylum, a phylum Ascomycota, commonly called the ascomycetes. And these are some of the fruit bodies of ascomycetes that you can see here, much more crusty, not fleshy fungi in this case. Although these are rather fleshy. This is the orange peel fungus. This is a cup fungus, or cup or saucer fungus. And in this case, the spores are produced on this surface here. They're produced in little sacs, usually or typically with 8 spores in a sac. They're called ascospores because these are ascomycetes. And here's a lovely imagery produced by Patrick Hickey, as was the last two movies that I showed. And this always makes me feel that it's like bonfire night, a wonderful firework display. To start with, you saw them, the spores being shot off, not to a great height, but when lots of them shoot off, they effectively form their own wind, which takes them up to quite considerable heights. So these are the spores shot out in groups of 8, almost like cannons. Okay, so those are some examples of fruit bodies, and certainly we've seen some diversity, but what are fungi? The fruit bodies are just the tip of the iceberg. Hidden within whatever the fungus is growing on or feeding on is the main part of the fungus, the business part of the fungus, called the mycelium. It's a network of tubes. Mycelium consists of an interconnecting series of tubes with rigid walls. They contain cytoplasm. They are microscopic. This is a micrograph that you can see here. I suppose these hyphae, the individual tubes of filaments are called hyphae. This is a hypha, singular. And I guess each of these hyphae is probably about 15 micrometers in diameter. So really quite small. You need a microscope to see them. You can see these lots of hyphae together are called the mycelium. You can see the mycelium en masse when it grows, as in this example on a plate of agar jelly in the lab. You can see it growing on the surface here. I should point out that it doesn't just grow on the surface, it grows within the whatever it's growing on. And this is one of the important things about fungi. These individual tubes, these hyphae, can grow into whatever it is they're feeding on. Each hypha grows from the tip. Sometimes side branches form further back to fill the gaps. Maybe we can, oh, that's a bit peculiar. Screen sharing has stopped. Well, we don't know why that is, do we? Should we try again? You might have to do some, oh, how strange, PowerPoint is closed. Well, that is a panic. Just hold on. You might have to do a little bit of editing here to this video. Let's just
no problem.
If we can open that again. Don't know why that has done that. Can't say that's happened before. Document recovery. Okay, let's minimize that and try to screen share again. Oh, it's not displaying, is it? Okay.
No.
Let's see what is going on. Okay. I'm definitely not an IT wizard. I have to tell you this, but don't panic that we had over. We just try and find the way back to where we were. There you go. There's just a quick recap for you all there. And I think we were somewhere around here, so we'll go back to there.
Looks about right.
Okay, we'll carry on from there. So we can see this again, this video with each individual hypha growing from the tip with side branches coming up to fill the spaces. Okay, I don't know what is going on here, why screen sharing keeps stopping. Can't say I've ever had this problem before. Are you seeing that? Okay, so individual hyphae, as I say, can't be seen except for with a microscope, but en masse you can see them. And here, individual hyphae have come together, they've aggregated, and you can see mycelium growing en masse across the screen. And here it is growing again. You can see them aggregating actually to form these cord-like structures. And we'll hear more about mycelial cords in a moment. So again, thanks to Patrick Hickey for this wonderful micrography. So moving on, you'll have got the impression, quite rightly, that mycelia grow outwards, and they often grow in circular patterns. And you can see examples of this in the natural world. So for example, here is ringworm, a fungus growing on somebody's finger. And here is a picture of a fairy ring, this time indicated by this dark green region of lush grass. The fungus would have started growing in the middle here and then grown outwards. And the reason we have this lush green ring is because in this instance, the fungus is actually feeding the roots of the grass. Sometimes you'll see fairy rings which comprise dead regions of grass. In that case, we have a parasitical pathogenic interaction between the fungus and the grass. And actually, sometimes you see lots of these fairy rings, and some of them can be really quite old, up to hundreds of years old, if you work out the speed with which the ring is increasing year on year. Here we've got another example of a fairy ring. If you get your eye in, I know it's perhaps a little bit difficult to see, but look, here is a ring of mushrooms, the fruit bodies of a fungus. And just to remind you that the mycelium is the main part of the fungus, here is that same species of fungus there fruiting, and there is the mycelium revealed for all to see. Now, I don't want to give you the impression that fungal mycelia only form circular patterns, because they don't. They'll form patterns depending upon where their food is. And what you can see here is the forest floor, and it's a photograph by Wendy Thompson, and which she obtained when she was doing her PhD. And she revealed remove the surface leaf litter. You can see it's a beech woodland. You can get an idea of the scale from this beech leaf here. The litter was carefully removed to reveal this network, this mycelial network, a network of mycelial cords. So these aren't individual hyphae, because remember, you couldn't see those with the naked eye. They're hyphae which have aggregated together to form these linear organs. So I guess this one is a couple of meters across, or that's how much of it you can see in the picture. Now, let me give you some idea about how these networks and how their patterns form. Now, you'll think that these photographs are really rather poor quality. Indeed, they are. These were photocopies. Now, what we've got here is trays of soil. This is a tray of soil, about 24 by 24 centimeters. This is the first experiment that we ever did on the behavior of cord-forming fungi. It's work done by Chris Dowson when he was doing his PhD. A block of wood was colonized by one of these cord-forming fungi. There it is. And you can see it starting to grow out. And these are the control trays. That's a plastic bottle cap, and the fungus grows outwards. It reaches the plastic bottle cap and, of course, ignores it. This is a wood decay fungus. It's searching for wood, and it grows out further. This is after about 80 days after the start of the experiment. You can see we've got a mycelium, and you can see these cords, these aggregations of individual hyphae. But let me show you now the situation where rather than a block of wood, rather than a plastic bottle cap, we had a block of wood, uncolonized wood. And this picture here is the situation after 80 days, so equivalent to this. So to start with, this was just a block of wood, like this block of wood grew out, but rather than meeting plastic bottle cap there, it met an uncolonized block of wood. So if we looked at the equivalent time, you would have seen mycelium growing all around there. But when it's met this wood, it started to thicken up this cord joining the two, and messages have gone to the mycelium that was here, effectively telling it that this is the successful search pathway. So it thickens up this region, this other mycelium dies back, the nutrients in it are recycled for further growth, and then you can see later on it's grown further in search of yet more food. Now, size is important. Look at the same situation, but this time with a small initial block of wood, and it meets a great big new food source, a new set of blocks of wood. And you can see that all of the mycelium that would have been around here has died back. And so you can envisage how these cords form on the forest floor, linking up between pieces of wood, twigs, beech cupules, etc., etc. And as rather a fun thing, at one point, we set up a time series of wood blocks with cord formers growing out of them in the form of a map of Great Britain here. And we sort of roughly given the size of the wood blocks that the fungus grew from was roughly equivalent to the population size in that area. So you can see London's got a huge, great big one. So that's in London. I expect this is Birmingham, Manchester. This is the north, representing the northeast, Edinburgh, Glasgow. This will be Plymouth down here. And they grow out. Fungi like you and I, when we can recognize we're individuals. When they meet a different individual, they don't fuse together. When they meet an individual which is the same as themselves, which is identical, genetically identical, which is self, just split up, they will fuse again. And you can see they've done this here. Look, there's a thick cord forming here between London and Birmingham. There's another one there between London and Bristol, and Cardiff plus Newport and Swansea. And you can see that the road network is forming. We have the M4 here. The M5 actually started to form, but seems to have died back there. No longer goes down to Exeter and beyond. I guess that's what's this, is that the M1 and then the M6 and so on. And of course, there are a few strange things that happen. So we've got quite a nice roadwork network in mid Wales, which of course doesn't actually exist because we haven't been able to incorporate hillsides and mountains. And likewise, something's gone a bit peculiar in Scotland because the fungus buried down into the soil and then erupted again further up. But that's just a fun thing that one could do. Now, I don't want to give you the impression that all fungi grow out of resources searching for new ones, because that most certainly is not the case. Probably most fungi are confined within limited resources. So we have a Mycena species on a twig here. We have an Onigina species here, which is using this animal horn. And this is the nail fungus Poronia punctata, which is a dung fungus. It's growing on a patch of dung. They don't grow out. The only way that they can spread is by spores, not by this mycelial spread. Though, of course, mycelium is ramifying throughout these substrata here, because that's what they're feeding on. And it's the mycelia that grow and feed. So on what do fungi feed? Well, if we take that huge kingdom, 3 or 4, 5 million species as a whole, then the fungi can actually probably live on every organic compound that there is. Fungi live on both dead organic matter and on living organisms. And there are 3 broad groups of fungi that we can distinguish, although these can blur into one. This is just 3 ways of feeding on a spectrum. So there are those that are called the saprotrophs, and they use dead plant and animal tissues. Then there are the necrotrophs, which kill plant and animal tissues and then use them during and after killing until they're replaced by other fungi. And then there are the biotrophs. They live on living cells and tissues. And I suppose immediately you might think, oh, they're the baddies then. And they must be parasites. They must be harmful. And some of them are parasites, such as the rust fungus that you can see here. But others, in fact, very, very many, are beneficial to other organisms. In terms of plants, they're mutualists when they form what is called a mycorrhizal association. And I'll turn to this in more detail soon. Mycorrhizal literally means fungus root, that you can see here. And then lichens, they are mutualisms as well. A lichen is a fungus plus photosynthetic partners such as algae or cyanobacteria. Let me tell you a little bit more about the saprotrophs, the decomposers. If we walk through this deciduous woodland, this part is largely a beech woodland, as you can see. We're absolutely surrounded by fungi. They are in all of these decaying leaves on the forest floor and decaying woody material, decaying logs. It is the fungi which really are the only things which can break down lignin on a reasonable time scale. And they do a really important job. You might think that they're a bit of a nuisance when they're breaking down materials that you don't want them to, like perhaps your fence posts, for example. But without them, the ecosystems of our planet wouldn't work. All the dead stuff we see have nutrients locked up in them. And if it weren't for the fungi decomposing this dead stuff and releasing nutrients, our ecosystems would soon run out of nutrients and there wouldn't be free nutrients available for plants to carry on growing. So they're doing an absolutely crucial job. And incidentally, and I won't be talking about this today, perhaps this is a talk for another time, but you can see all these lines in this beech log here. These lines are demarcating the territory of individual fungi, a bit like us with fences or walls around our homes and gardens and medieval cities. So that's the territory of one individual in there, another one there, another one there. So this could be different individuals of the same species or different individuals of lots of different species. And they are having a white battle there. They protect their territory and they fight. As I say, that's for another day, perhaps. Now, I talked about another way of getting food, and that is as necrotrophs, killers. And there are fungi which can kill both plants and animals. And indeed, they are a worry to us from the perspective of obtaining enough food to feed an ever-increasing population of humans. And indeed, some of the main plant pathogens, which are wheat stem rust, rice blast, corn smut, soybean rust, and potato late blight. I should say that potato late blight is not caused by a fungus, but by a fungus-like organism, something a bit like fungi, but it isn't a fungus. Anyway, these diseases cause enough loss of crops, which would feed something like between 600 million and 4,000 million people every year. So fungi as pathogens, killers of plants, are important from a human perspective. And they do kill our trees. I put as the example here, ash dieback. You can see these are shoots of these ash trees that have died here. These ones are still okay. You can see the classic girdling on the stem, and here are some of the fruit bodies of these ascomycetes on the petioles, the rachises of the ash leaves on the forest floor. One thing I should say, though, that we tend to blame fungi for killing whatever it is that they're killing, and of course they are doing that. But I should say that more often than not, the blame lies squarely at our own feet, mankind's feet, because we move plants and animals and the diseases they carry around the world. It is us that moves them to new places. It was us that moved, for example, ash dieback across Europe, and there are very many more examples of this. And of course, it's us that grows our crops in these vast swathes of monocultures, which allow a fungus, when it can cope with a particular plant's defense system, it can then just rampage through whole crops. Okay, so that's, from a human perspective, a bad thing that fungi do. But let me tell you this. It was fungi that allowed plants to colonize land 500 or perhaps 450 million years ago, something like that. And these are just some fossil plants that you can see here. And as I've already intimated, fungi form important mutualisms with living plants. These are called ectomycorrhizal fungi. Now, ectomycorrhizal fungi form an association with the roots of trees. Now, this little pine seedling, it's in a little microcosm, I don't know, about 20 centimeters or 25 centimeters, something like that, from top to bottom. This is the root of the pine, and it goes down there and along there, more root across there and a bit around there. All of this that you can see here, that is fungal mycelium. Now, actually, there is no root really in contact with the soil. So although we were taught at school that it's the roots and the root hairs which soak up water and nutrients, more often than not, it is fungi that do that. In this case, it can only be fungi because there's no root in contact with the soil. Here's another example here. You can see a thicker bit of root that may be in contact with the soil, but that's not an absorptive bit of root anyway. Now, here is a mycorrhizal root tip. So mycorrhiza literally means fungus root. What you can see is this root tip, it doesn't have any root hairs at all. The fungus is completely sheathing it, in this case, and growing out into the soil. It soaks up water and nutrients, sends them back into the plant, and in return, the plant provides sugars, which is made during photosynthesis. So it really is a mutualism. And this is going on underground. This is a lovely piece of artwork produced for me by Mariana Heilman, who really got to grips with and understands what's going on with these mycorrhizas. Now, you could imagine that trees, they actually have lots of different mycorrhizal associations with their roots. So imagine here we've got just perhaps two species, although there'd be many more, a purple species and a green species attached to the roots of this fungus. And you can see here their underground network. You'll notice that this green fungus actually also attaches to this next tree and the next one and the next one. On the other hand, the yellow fungus, that's only joined up with two trees, and so on. And you probably heard of the wood wide web. This is this network which we see below ground, a network of these mycorrhizal fungi, which feed plants and indeed sometimes can move nutrients or sugars from one tree sometimes to another, or indeed sometimes messages from a tree pass along the mycelial interconnections to a neighbor. And it's not only trees that have this association. In fact, 90% of plants do. Most herbaceous plants have what are called arbuscular mycorrhizas. This time, the whole of the root isn't ensheathed, and indeed, the roots do have root hairs, but the fungus gets inside the root, indeed, this time inside the root cells, which is what you can see here. It's stained red, this particular fungus. And this is where the swap occurs. Water and nutrients provided by the fungus, the plant in return provides sugars. Ericaceous plants, heathers, rhododendron, things like that, they also have mycorrhizal associations. They're called ericoid mycorrhizas. They're a bit different. Here's a root, and you can see these outer cells. The blue stained region, that is the mycorrhizal fungus. You can see the hyphae growing out into the soil. Same basis of the mutualism. Fungus provides the plant with water and mineral nutrients, and the plant provides sugars. If you look at these cells carefully, you can see that about 80% of the cell is taken up with fungal mycelium. And yet this is a mutualistic relationship. And to show you why it is fungi which actually are really effective at getting water and nutrients, this is a drawing that I've made of some roots of arbuscular mycorrhizal plants. I know that because these roots have root hairs. Remember, the ectomycorrhizal plants don't have root hairs. So here are the root hairs, which take up water and nutrients. And this yellow region is the nutrients that are available in the soil. But of course, the root hairs have taken all of the nutrients in the vicinity of themselves, and they can't get at these other nutrients. But look at the situation where there's a mycorrhizal mycelium. The hyphae extend well beyond this zone of depletion into other regions of the soil, and indeed can get nutrients from everywhere. So that's why they work so well. They're much finer than root hairs, and they can spread a long way. Now, there are some other relationships, sometimes called mycorrhizas, sometimes called mycoheterotrophs, some relationships in which, well, let's put it this way, there's cheaters in all walks of life, aren't there? And this time it's plants who are the cheaters. These are monotropa plants, Indian pipe, and they don't have chlorophyll. They do have fungi associated with their roots. They are fungi such as russula species and Suida species. These species of fungi actually form ectomycorrhizas with forest trees. Now, the fungi again provide the plant with water and nutrients, but they don't get sugars in return. These are cheaters. They don't produce chlorophyll. They don't photosynthesize. And the plants actually get sugars from the fungus. The fungus is getting these sugars from the trees. So effectively, the trees are being parasitized by this plant via a fungal link. Orchids too have associations with fungi. As you know, orchid seeds are very tiny, and they have to form the mycorrhizal relationship. Otherwise, the seeds are not able to grow into plants. Now, some orchids do form chlorophyll when they grow up. To start with, they don't photosynthesize, and again, the fungus has to provide everything. Those which do produce chlorophyll when they've grown up can effectively pay, in inverted commas, for their water and nutrients by giving sugars in the same way as I've already described. But again, some of them are achlorophyllous. They don't produce chlorophyll, and again, they are acting effectively as parasites on the fungus, getting the sugars from the fungus too. And as I mentioned near the start, lichens are a partnership of fungi with algae or with cyanobacteria. That, of course, is where we get the green color from. Here's an example of a lichen thallus. So it's a sandwich, really. There's a layer which is fungal, another layer which is fungal, and a more open layer of fungal hyphae here. And then there are layers of the photosynthetic cells. And the basis of the mutualism is almost the same as with mycorrhizal associations. Fungus gets the water and nutrients which are deposited on it, or which the mycelium can get, and the algae or cyanobacteria produce the carbohydrates. Now, you know, I'm very often met with the reaction, oh, when I start to talk about fungi, oh, yuck, what a nuisance they are. And people will tell the story of how they rot our crops and our homes, and indeed our food, as you can see here. But I'd like to tell you from a human perspective how important they are. I've already, I hope, explained to you that fungi are crucial for our ecosystems. One, because they're the decomposers and they break down and recycle things so that plants can grow. And two, because 90% of plants have these mycorrhizal associations with their roots and would not exist without them. So our ecosystems wouldn't work without fungi. But we as humans really need them directly as well as indirectly. And indeed, for thousands of years, for 5,000 years or more, and probably much more, man has used fungi not just to eat. This was Ötzi, who was found, I think, in the Italian Alps. I think he'd been stabbed in the back and then preserved for these 5,000 years. And he had a sort of a necklace, which this is a reconstructed one that you can see here, made of this fungus. Used to be called Pictoporus betulinus, now it's called Fomitopsis betulinus. He also had Fomes fomentarius, I think, in his little bag. And so ancient man used to use fungi too, sometimes for things like sharpening tools or for starting fires, or indeed as medicines or for staunching wounds or what have you. Of course, we eat fungi because fungi are very nutritious. They're high in protein, low in fats. Indeed, many animals eat them, including vertebrates eating fruit bodies, and many invertebrates depend directly or indirectly on fungal mycelium as a food source. Of course, we know about making alcohol, that the yeast make alcohol. Yeast are fungi. Of course, they make bread rise, and you can have yeast extract too. They also are important in soft drinks. They produce citric acid in great big vats, and this citric acid is used in all canned drinks, soft drinks, and in jams and confectionery, canned fruit and vegetables very often too. It's an acidity regulator. Rather than having soy protein, there is fungus protein that you can buy. They're also important in chocolate, in making chocolate. Not actually the process of making chocolate, but when the cocoa pods are harvested and they're broken open, and there's this gelatinous mass with the cocoa beans in, and you have to get the right fungi growing on that gelatinous material, which gives the wonderful flavor to chocolate. Cheeses, of course, blue cheese is an obvious one because you know that those are fungi which grow through, but soft cheeses as well are products of fungi. And indeed, most of the hard cheeses which we buy in all of our supermarkets, and I should say that all of these brand names, there are very many other products too from other suppliers that you could purchase. But in hard cheese, most of the hard cheeses which we buy, they have a fungal product to make them. So you know that the enzyme rennet is used to make cheese, and that used to be obtained from, I think it's the stomach of calves, but now it's again made in these vast vats. It's a fungal enzyme. That's why you often see cheeses often say that they are suitable for vegetarians, because it's a fungal enzyme. Now, I've mentioned before that I talked about fungi being killers of plants, but there are fungi which also kill animals, in invertebrates, for example. So this one here, the common name is green muscadine, Metarhizium anisoplii. And you saw wriggling around in the bottom there, a nematode that had been trapped. In this case, it's called green muscadine because although the mycelium is white, the spores are green coloured. This is a pea aphid, and here are the spores which have been shot off this pea aphid. You saw nematodes being trapped. Some nematodes are pests of plants. There are lots of invertebrates, insects and what have you, that are a nuisance to us from a farming perspective, feeding on our crops, devastating our crops. And some of these fungi at least can be used as biocontrol agents. Indeed, this fungus, there is a commercial formulation of it to use as a biocontrol agent rather than having to use nasty pesticides. From a human perspective, or again, an animal's perspective, fungi do grow on us sometimes, and indeed can cause problems. Things like athlete's foot and ringworm can relatively easily be cured by topical application of medicines or ointments and what have you. But I'd just like to draw your attention and remind you that fungi produce some very important drugs, the wonder drugs of our age, and the obvious one that springs immediately to mind is penicillin, produced from the fungus Penicillium. Indeed, there are quite a lot of other antibiotics from fungi. Statins that control cholesterol, they were discovered in fungi. Cyclosporin, another fungal product, it's used to prevent transplant tissue rejection. Ephedrine treats asthma. Ergot alkaloids are used as vasoconstrictors and migraine treatments and to induce uterine contractions. And they're also used in the production of some steroids and anti-inflammatories. So they're very important from a medicinal perspective. And of course, many cultures use herbal remedies and fungal remedies. And I expect that some of these certainly are efficacious, although we need to do a lot more actual proper clinical trials to know what compounds can actually be used in our modern day Western medicines. But I have absolutely no doubt that there are many, many more medicines out there still to be found. Fungi are also important from a bioremediation point of view, or from a recycling point of view. I've talked about natural recycling, but do we think about the mess that we've made of our planet with toxic metals and contaminated soils, plastics, pollutants of one sort or another? There are fungi which can begin to remediate some of these situations, such as organic pollutants. Fungi have been found which can digest plastics, and hopefully in the future we will be able to use some of these rather more than we do now to help clear up some of the mess which we humans have made. But I'd like to finish off by reminding you that without fungi, planet Earth would not work, because plants would not be able to grow. Plants fix vast amounts of carbon every year. They are the powerhouses of our planet, but they do require water and nutrients. The decomposers recycle nutrients, and the mycorrhizal fungi help to take these up directly as well. And I would also thank all of those who have provided images which I've used, and also the lovely video clips provided by Patrick Hickey. So on that note, thank you very much.
Thank you for that, Lynn. That was brilliant. Thank you so much. We have a couple of questions in the chat if you want to have a look. And if anybody else has any more questions, please do pop them in the chat.
Right, I'll try and start at the top if I can shoot up there. Let's see where we go.
The top one is how fast is that growing, which I think referred to the video that you showed.
Oh yes, okay. So I showed several videos, didn't I? Right, to give you right. Fungi grow at different speeds. Some can grow very quickly, and indeed there are some fungi which can grow so quickly you can actually see them growing under the microscope. Very good for student practicals. If we were thinking about, for example, one of those agar plates that I've got, I suppose that probably again, it varies between species, but they will perhaps grow a few millimeters a day, sometimes up to a centimeter a day across those plates. But it is variable depending on the species and depending on the conditions as well, whether you're under optimum conditions. Oh, so here's the question. Is it impacted by weather? Are there conditions where it does not grow so well? Absolutely. So obviously fungi grow colder, grow slower when it's colder than their optimum. So many fungi have optima for growth at around 25 degrees C. There are fungi in polar regions which have much lower optima. There are some fungi which have higher optima. Is global warming impacting at all in a positive or negative way? Global climate change and indeed many other aspects of environmental change are impacting hugely on fungi and on ecosystem processes to which they contribute. So for example, if we were just thinking about the fruiting of fungi, pre about 1978 in southern England, the autumn fruiting season. So from the beginning to the end of when we are seeing fungi fruiting, obviously that they fruit, different fungi tend to fruit a bit at different times, but the average length of the fruiting season was something like 33 days. And now it's something like, well, it's over double, that's 70 something days. So the fruiting season has changed. And you might think, oh, well, that's only of interest to anybody who's interested in going around finding out, identifying fungi, picking fungi and whatever. But it really is telling us that our ecosystems are changing and behavior of the fungi is changing if they can now fruit for that long. There are other differences too. So for example, mycorrhizal fungi tend to very often now to fruit on into the winter. And if you look at trees, where are we now? We're, what is it, 17th of November, well through November. When I was a child, the leaves were off the deciduous trees before bonfire night. Often in December, we'll find trees in leaf, oak trees in leaf in December. So you can imagine they're still feeding their mycorrhizal partners sugars, and the mycorrhizal partners will still be feeding the trees while they're still photosynthesizing. We now find fungi which fruit in the spring, which never used to. There were always some fungi which fruit in the spring, but lots do now. They can fruit in the spring and the autumn. Now that's telling us something that's changed in our ecosystems. I think it's telling us when it's decomposer fungi, the saprotrophs, that the winters are much warmer and those fungi are active in the winter and they're able to accumulate nutrients sufficiently so that they can fruit in the spring. So there are all sorts of changes going on. I touched very briefly, but said it's a talk for another day, about fungi they fight each other. I showed the picture in wood with the little interaction zone lines which demarcate their territory. Well, communities of fungi largely change because of these battles that you get between fungi, and we found that at different temperatures or different amounts of water, this changes the outcome of those battles. And so you'll be getting different communities of fungi. We know that the pathogens of crops are moving globally. They're moving sort of northward. They've got bigger ranges, different ranges now. So there are very many things which are changing because of climate change and other aspects of global change. You mentioned, are pesticides and fertilizers impacting growth in any way? Fertilizers they have a major effect. I mentioned flower-rich hay meadows, which are the so-called unimproved grassland because fertilizers have not been added. When fertilizers are added, you lose a lot of fungi. So our normal grasslands are lacking lots of these rare taxa because of fertilizers being added. When you get nitrogen pollution, be it from dropping in from the air or again from fertilizers, you get differences in the mycorrhizal fungi associated with the roots of trees. For example, you lose a lot. Some others thrive, but there are huge changes going on because of what we as mankind have done. Somebody says, I think that honey fungus seems to be selective about what it kills. Why is that? Well, actually, you know that there are several species of honey fungus, and they're not all killers to the same extent. So the one which really goes for a wide range of different plants is Armillaria mellia. But there are other fungi which form rather more of these bootlaces, for example, Armillaria gallica, which does finish off suppressed plants, so plants which are in a bad state or light suppressed trees, but probably doesn't go for things which are healthy at all. So that's a different species. There's another fungus, Armillaria ostoi, which is a killer of pine trees. So when you get fungi which are selective, it depends upon their mode of attack. So some of the pathogens, some of the biotrophic pathogens are very selective. The biotrophic pathogens of, we didn't talk much about this evening, although it was the subject of actually one of my undergraduate lectures today. Biotrophic pathogens are very selective very often for the plants which they parasitize, because over evolutionary time, they've evolved to cope with the defense mechanisms of the plant. It's a very subtle relationship. So it depends on the mechanisms to some extent that fungi use. Question, can you talk a little more about ash dieback? If you'd like me to, I can. So yes, the ash dieback fungus, Hymenoscyphus fraxineus, which is an ascomycete fungus, colonizes leaves. It may, in some cases, it gets down the petiole into the stem, and then if it gets into the stem, it can girdle the stem, especially if the stem is a young one, so fairly small. But it doesn't always do that. The leaves fall to the floor, and in the early summer, spores are ejected. Their ascospores are ejected from on the floor, and then they somehow find their way into the canopy. It was a disease which originated in Asia, and in the forest in Asia where it's native, it doesn't really cause problems at all. And this is a classical thing about pathogens, where they come from, where they've evolved over probably many years, often hundreds, thousands, or millions sometimes of years. The plants and the fungi are in balance, if you like. It's often when they are taken out of their native area and they find what we call naive hosts, in other words, plants which have not experienced the pathogen, that we tend to get problems. One of the difficulties with ash dieback appears to be that, and this hasn't been researched awfully much yet, is that Armillaria, sometimes you get basal lesions, as well as all this stuff going on in the canopy, you get these basal lesions where where you'll find both the Hymenoscyphus fungus, but also it seems that you get an Armillaria species in, and so it looks like it may be that Armillaria can also be weakened in the tree, and then perhaps causing problems where trees might come down. But we need to do a lot more research on that. I'm lucky enough to have a student who's working jointly with forest research at Alice Holt, who is looking at some of these aspects. Are we likely to lose all our ashes? Hopefully not. The ash tree in my garden is still doing very well, and I live in the Wye Valley, and the Wye Valley has been hit very hard by ash dieback, but my ash tree is still surviving despite being surrounded. I mean, I think there might be for localized trees in your garden, that one thing that you probably might be able to do to help matters is, if you do get infected twigs and leaves. When the leaves fall to the floor, they go very, very black, and you get the little fruit bodies produced on those in the summer. If you get rid of those leaves, you're getting rid of that source of ascospores. So it may be the possibility, and I have done that. Well, I've removed made sure the ash leaf litter isn't there. That may be something which we could do on a sort of a very localized scale. What was the name of the paper demonstrating mycelium growing towards wood blocks? Well, that paper that I showed you, I think it perhaps wasn't on the screen in this instance. I usually have that on the screen. It was a paper by Dowson, Rayner and Boddy in 1980 something, maybe 1986, published in the Journal of General Microbiology. We have subsequently, ever since then, worked on the foraging activities and the behavior of these fungi, and have written quite a lot more papers. If you're interested in anything more on fungi and trees, I'm going to take this opportunity and be very cheeky and advertise a book that I produced recently. It came out in, I can't remember when, March this year, called Fungi and Trees, Their Complex Relationships. And it is written for a general readership rather than an academic audience with, I'm flicking onto pages that don't have many illustrations, but it has loads and loads of nice pictures and illustrations. That might give you some more information. And on another level, another book that I was lucky enough to be involved with recently, that I wrote the text for, it's a Dorling Kindersley book. It's aimed for children aged 7 to 9 year old, but I tried to make sure that it was also really useful for adults who are reading it with the kids, called The Humongous Fungus. And again, it's quite nicely illustrated and actually gives you quite a good way into the ecology of fungi and the importance of fungi. So cheekily, just a couple of adverts there.
Just aware we're coming towards 1:30, Lynne, so if you want to maybe pick one or two more questions.
I think probably there is only one actually, so we can whiz through that probably in 2 minutes. The question says, I have heard that plastic can be broken down by fungi. Could you expand on this? Well, I don't actually know very much about that to expand upon, I'm afraid, but there are examples of a few species of fungi which have been found to break down plastics of various sorts. And I dare say that as we expand, when I say we, that's the fungal biology community, I guess, rather than myself personally. I imagine that we will find fungi which can break down plastics, and hopefully will make inroads into the wretched mess that we've made of things. But I think that in the first instance, we really have to reduce our plastic use and not to carry on in the way we have, because clearly things are not sustainable. And I think that probably is the last. Oh, I don't know. There's some more which might have dropped off my page.
There's lots and lots of questions, but I'm not sure we're going to have time for them all, unfortunately.
Oh, golly.
Yeah.
What is the role of the fungi which is poisonous to humans? There are lots of fungi which are poisonous to humans. Whether they evolved to inhibit invertebrate grazing, I don't know, because some of the really toxic things can still be grazed. Can you tell us more about the marine fungi? I don't think we've got time to think about that. There are fungi in all environments on the planet. I'm always disappointed when I see people have just kicked over fruit bodies. Why do they do it? I don't know why they do it. Does that stop them from producing spores? It stops them from producing spores, yes, because they've been severed from their mycelium. So it does prevent spread of the fungus. The mycelium that's underground is still okay. Yes, fungi have been used to replace some leather products. Indeed, for a long time, people have sometimes used some of these woody brackets to make leather. But of course, they've been leathers in inverted commas are being made in another way by mycelia now. If you have fairy rings on your lawn, does mowing do harm? No, mowing doesn't do harm. The mycelium will still be there underground. Might be nice to leave the fruit bodies while they're fruiting so that they can spread. Does relocation of soil work? I think that maybe that's a question that I don't have time to answer. There's a plastic one, we've mentioned that. Should we be concerned about estimated 60 years of topsoil left in farming? Yes, we should be very concerned, and we have to find ways of farming better so that we're not losing our soil, and indeed sometimes different types of farming. There's a very good video, I think, available by Bridget Emmett. You might be able to get by Googling it. She gave a talk at an Arboricultural Association meeting a couple of years ago where she gave a fantastic talk about soil. I'm aware that we've gone over the 7:30 time. I can carry on trying to answer if you want to, but I do fully understand if we should stop.
Do you want to just pick one more to answer maybe, and then we'll call it there? I think there might be only one left. You've shot through the last few there.
Oh yes, I did shoot through, and I missed some of them. Oh, there's lots of nice thank you. So maybe it's maybe that's a good time to stop.
Okay, no problem. Well, thank you so much for that, Lynn. That was absolutely fantastic, and thank you to everybody who attended for your brilliant questions. I will send out a recording of this event in a post-event email, probably tonight or tomorrow. So you'll have the recording to watch back if you want to do that. So I think we'll say good night. Thank you very much, Lynn.
Thank you.
Thank you, and goodbye, everyone.
Bye.